So the nice thing about the Peter Wally Institute and these colloquiums abroad is they also fund 3 speakers from UBC to come here. So Bill is the first of 3 UBC speakers, besides me, that will be speaking at this meeting. And what's nice about Bill is that he's an environmental microbiologist, and he's studied the diversity of microbes long before people in health and things ever really thought about it, and up in the oil sands and the Arctic and various other places. And so I remember when I started talking to Bill about maybe looking at gut microbiota and everything, and he was delighted because actually the gut microbiota is very simple compared to the world that he works in out in the environment. So Bill's been very heavily involved in developing tools and technologies to analyze microbes and try and make sense of this incredible diversity we see, and he's been helping our lab very much in this area. So introduce Bill Mohn from the University of British Columbia. His title is A Hard Look at How We Look at the Microbiota. Bill.
Thanks, Brett. I really appreciate the opportunity to participate in this colloquium. Et je remercie nos hôtes d'avoir organisé ce colloque. So I'm one of the people Brett mentioned who is has a newcomer to this field, as he said, and I'm going to be focusing on some of the methods we use to look at microbiota. And those methods have come a long way since this then Leeuwenhoek microscope was state-of-the-art. So another version of the title would be a microbial ecologist's perspective of recent advances in understanding microbiota. So I'll give you a very general overview of molecular microbial ecology, focusing on many of the benefits and limitations of this general approach. And then I'll also include some examples of various approaches to analyze microbial communities. Now I know some of the audience is probably very familiar with techniques that I'll be talking about. Others are maybe used to seeing some of these analyses, but maybe don't have such a background in them. So I think it may be useful to just break it down and to look at what we can and cannot learn from some of these approaches. And for those who are much more familiar, hopefully maybe some of these analyses, largely taken from macroecology work, might be of interest. Okay, so the whole idea of molecular microbial ecology really stems from this basic problem, the Great Plate Count anomaly. And so we've known for a long time that we can only culture a very small proportion of the viable cells that we can observe in just about any natural environment. And so that led to this basic strategy. With the advent of molecular biology, microbial ecologists saw a real opportunity, and it's certainly fair to say that this has revolutionized the field of microbial ecology. And so what I've tried to do here is, in a very simple way, summarize the basic idea, and that is to to sample DNA from a microbial community and use that as a proxy, and to then analyze, using various methods, the DNA to learn more about the community. And some of you may remember this analytical method, but it's not in much use today. So I'll talk about some other ones instead. So first, I just want to focus on this very basic idea of looking at the DNA as a proxy for the community, because it does have a number of important benefits and limitations. And so it solves this basic problem of not being able to culture organisms. And so that's great and probably the biggest benefit of this approach. Another point is that the sample is homogeneous. And so this is very useful for being able to get a representative sample of a community that may be very different over small spatial scales. So that's another big advantage, to be able to turn it into a solution that one can then just simply analyze. But that has a trade-off, and that is that you lose any kind of spatial information when you take this approach. So we don't know which cells are directly in contact and likely interacting with which, or with their substrate, and of course in the case of the gut, the host animal. Another advantage of this approach is that you can get very good taxonomic information from the DNA, of course, But again, the trade-off there is that you lose information about the physiological status of the organisms. So you may, based on the DNA, know something about what they can do, but you don't really know the status of the cells when you extracted the DNA. And another big benefit is that the DNA analyses are amenable to a whole range of analyses, including high-throughput sequencing, the use of databases that have become very extensive, and bioinformatics software. And so this is really, I would say, a second revolution in microbial ecology is the advent of genomic approaches. And so that, again, has been a real leap forward in our capabilities. And the last point I want to mention is that this simple act of extracting the DNA does not come without some bias. And this is really important to keep in mind. So what we see will depend on how we extract the DNA. And I think of this as a spectrum. And you kind of have two options. You can either use a fairly gentle method or a much more intense method to try to extract the DNA, and especially with respect to any kind of physical method to lyse the cells. So if you take the gentle method, which you need to do for some applications if you need large pieces of DNA, you'll minimize the shearing, but you're also not going to lyse all of the cells, for sure. And so there's going to be a bias against cells that have tough cell envelopes, and so organisms such as some of the actinobacteria and others will probably be biased against, or I would say will be. On the other hand, if you use a more intense extraction, you're going to accomplish the disruption of almost all the cells, but you're going to shear the DNA, and especially you're going to shear the DNA that came out of the cells early on in the extraction process. And so then you've got the opposite bias against cells with less robust envelopes. And so some of the proteobacteria and others would be biased against. So this is an important trade-off. And one possible way to address this is through sequential extractions. And so the basic idea being do a fairly gentle extraction, get the easily obtainable DNA out, and then re-extract more with a more intense method, and you could do one or 2 or 3 rounds. So this, in my experience, has definitely improved recovery of DNA from diverse organisms. But of course, this is very laborious. And so if people are looking at large numbers of samples, it may not be practical. And so one doesn't see this very often. Okay, so I want to just show you an example of how extractions can bias the DNA that we get out. And so this is a comparison. These are microbial communities from mouse feces that have been analyzed. And we're comparing 2 extraction methods. One is a commercial soil extraction kit and a fast prep tissue homogenizer. The other is a stool extraction kit and a mixer mill extractor. And so these are 2 of many options. And the way we've compared here, this is probably familiar to many of you. This is TRFLP, or terminal restriction fragment length polymorphism. And all you really need to know is that each of these peaks represents a population, or we might refer to it as a phylotype or operational taxonomic unit. And I'll use this abbreviation OTU throughout the talk. And then the other point is that the height of the peak, or the fluorescence intensity, is related to the abundance of that population, or at least the abundance of its DNA in the extract. And so you can see some real differences here. These are averaged chromatograms, or averaged fingerprints for replicate samples of using each method. And you can see very significant differences in the peaks that are visible. So it's actually quite a coincidence. The exact same number of peaks, 66, were detected by both methods, but there were a total of 68. So different peaks are detected by different methods of extraction. And also you can see some of the peaks that come up in both methods do vary significantly in their intensities. So you get a very different picture of the community just on the basis of the extraction method you use. And so one approach to try to minimize this effect is to test different methods. And so we've typically, in my lab, when we start working on a new community, we'll try a number of extraction methods and simply empirically look for the one that gives us the greatest number of OTUs, with reasoning that that's probably giving us the greatest representation of the community's diversity. But clearly, any way you go about it, you're going to bias your DNA extracts just through the act of extraction. Okay, now once you've got DNA to analyze, there are many, many, many different ways you can go about that, and I'm only going to talk about a few, actually just mention a few, and focus on one of these approaches. There's one major decision, and that is whether to directly sequence DNA or to PC amplify some target gene, usually the small subunit rRNA. So with these target amplicons, you can then go on and analyze them either by the fingerprinting methods, such as the one I just showed you, or more commonly today through sequencing, for example, pyrotag sequencing with 454 technology. But another very different option is to directly sequence the DNA in this mixture, or to do shotgun sequencing. And that yields a metagenome, the sum genome of all the organisms represented in the DNA mixture. And so that's really giving you something quite different. And so these two approaches have very different strengths. The PCR-based approach is best at telling us about community structure, or which organisms are present and what are their relative abundances. On the other hand, the metagenomic analysis can tell us about genetic potential of communities because it includes all of the genes in this metagenome. And so it gives you functional information about the community. So very different in what you can get from these two approaches. And I'm not going to talk about metagenomic analysis. I had thought there was going to be a talk on this before mine. But anyway, I just wanted to make a few comparisons before I focus on PCR-based approaches. So a couple of the strengths or benefits of metagenomic analysis, as I mentioned, the metagenome contains vast information about metabolism and other important processes, things that the community can actually do that may be ecologically relevant or have relevance for human health. Another important point is that since you do not do a PCR reaction, you won't have any of the biases associated with PCR. And so this could be an important factor as well. On the other hand, there are some limitations. One of the big ones is simply sample numbers. The amount of sequencing required for metagenomic analysis and the complexity of the analysis almost inevitably limits the number of samples that we can analyze. So it's fairly difficult to do this sort of analysis in a robust way with lots of replication, such as one often wants to do when looking or comparing microbial communities. Another important point is that the complexity of the bioinformatics and the limitation of our current annotation is a real impediment to metagenomic analyses. I also work on microbial metabolism, and I've looked at different types of metabolism over the years, mainly catabolism. And I know from experience that the annotation of genes in the various databases can be extremely misleading. And so this is going to be a problem for metagenomic analyses. I'm sure that we'll make a lot of progress in addressing these, but at present, these are definite limitations. Okay, so now I'll turn to the PCR-based approaches. And again, I just want to start by mentioning some of the benefits and limitations. So one of the important things about the PCR approach is that you're amplifying a single gene or gene fragment. And so these are very comparable and give you a very good definition of the OTUs that you want to compare among different communities. Another important point is that since you're using PCR, you can have a relatively small amount of DNA to start with, which can be important in some environmental samples. Okay, on the other hand, there are a number of limitations as well. One of the first ones is amplification bias. So this is when you end up with a relative abundance of amplicons that didn't represent the original mixture. Personally, I think this is something that people worry about more than they should. I think there are much bigger problems. Whenever we've looked at this in my lab, we've found amplification bias seems to be one of the lesser problems, but it's certainly possible. Another possibility is PCR error, and so that can involve misincorporation or chimeras, and the chimeras can be a real problem for some of these analytical methods. And finally, primer bias is maybe the biggest factor here, and so that's going to dictate who in this community is detected. There are a wide range of so-called universal primers that have been developed, and people have made a lot of progress in this over the recent years. But still, the fact is, whatever primer set you choose to analyze a community, you are going to target certain organisms and you're going to miss other ones. This is something unavoidable. And so this is going to be an important bias in any PCR-based method. All right. So I want to give you an example experiment and then look at some of the different analyses of the data. And I'm not really going to focus on the story of the experiment or the conclusions, but mostly just looking at the data. So this is a colitis induction experiment with 2 mouse strains, rag mice, and then rag mice additionally with a CD45 knockout. And colitis was induced by T-cell injection. And so you can see here what happened to the mice over time in terms of weight loss. So the control uninduced mice of both strains actually gained a bit of weight over the course of the experiment, whereas the ones with colitis induction after 2 weeks started losing weight. And so these were sampled then over the various time points in this experiment. So 4 treatments. And first I just want to give you a comparison of these 2 methods I've mentioned, fingerprinting analysis versus amplicon sequencing. And so this is for the experiment I just showed you. The data are shown in ordinations, and the ordinations in this case are via principal coordinate analysis, and this is an unconstrained approach. So basically there are no a priori assumptions about the groups, but rather this algorithm seeks to plot the different samples in a way where the most similar samples are closest together. And so similar samples should cluster in 2-dimensional space in this case. And this is a way of reducing very complex multi-dimensional data sets. Okay, so you can see on the left the TRFLP data, and this is based on not so many OTUs, just 20 to 29 OTUs or TRFLP peaks occurring in each treatment. On the other hand, pyrotag analysis of the same samples is shown here, and this is a data set with almost 300,000 quality reads and 3,000 to 5,000 reads per sample. And so there are more than 10 times as many OTUs, or in this case tags, sequence tags, in each treatment. So it's a much more robust data set. And you can see a big difference in the ordinations. Clearly, with the greater amount of data, it's possible to see the clustering of the different treatments in this experiment, whereas over here, that clustering is nowhere near as evident. Okay, but it's important to keep in mind that ordination is mainly a means to explore your data, and it's not a test of significance. So actually, when we do a significance test, in this case, analysis of similarity, this is a way of actually asking whether there are significant differences between these experimental treatments. And you can see that although the ordinations aren't as pretty, the TRFLP actually is capable of distinguishing treatment effects almost as well as the pyrotag analysis. And so these anisim results, you have a p-value indicating the significance of the treatment effects, and you can see treatment effects both for the different mouse genotypes as well as for the colitis induction treatment. And the R value is measure of the magnitude of the effect. And so we actually have a much bigger effect of the mouse genotype versus colitis induction in this case. And as I said, the differences between the two data sets are really not that great. So TRFLP is actually fairly powerful for seeing the differences. And another way to analyze these data is to try to break down the sources of variance, the things that are accounting for these differences in treatments. And permanova, or permutational analysis of variance, is an approach for this. And so in this case, you can see different sources of variability, the genotype of the mouse, the induction of colitis, and also time, so the different time points being compared. And again, the two methods are quite consistent, or the TRFLP versus the Pyrotag methods are quite consistent in what they show us. They show us that the genotype explains the greatest proportion of the variance and is highly significant. Colitis induction is responsible for a lesser proportion of the variance, and then time, a much smaller proportion. And this is the one place where it's different. And I have no idea why there's a gap here, but the table looked much better when I viewed this on my computer. But what this should be reading then is, well, it's just spread out this line and put a gap in here. So anyway, time is the one thing that the TRFLP analysis wasn't powerful enough to pick up the effect of time to show that it was significant. Okay, so that's a comparison of these two methods and also some of the key ways where we can look for treatment effects. And I guess the take-home message is really that the fingerprint data are much less complex and simpler to analyze than Pyrotag data, which can be a benefit, and are relatively powerful to measure community similarity. Having said that, I think sequencing has pretty much eclipsed DNA fingerprinting at this point because it has become so much cheaper and more practical than it was even a year or two ago. Okay, so the amplicon sequencing approach then, shown here, again has some unique benefits and limitations. So one of the important things is you have this superior depth of coverage. That is the number of OTUs that you detect, which we saw a moment ago. And so this may resolve relatively subtle trends in the data. So it could be very important in some cases if we're trying to discern, say, a treatment effect in the face of large variances, say variances among individuals, trying to get at, maybe what are specific treatment effects in an experiment. As well, the sequence approaches benefit from the extensive databases for small subunit rRNA genes that are now available, which allow really good taxonomic identification of groups. So a big advantage here is that we know who is responsible for the differences in communities that we detect. Okay, the downside here is the occurrence of sequencing errors. And so whenever you employ these methods that involve very large amounts of sequencing or of reads, there will inevitably be a significant number of erroneous reads. Okay, so sequencing errors can be quite significant in these data sets, and that has to be accounted for when interpreting the results. So the large data sets that result from things like Pyrotag sequencing require some kind of a pipeline, as people like to call it, for analysis of the data. And some of the basic functions of these pipelines are first quality filtering of the sequence data, and then binning. So this is actually dividing the individual reads into the OTUs. Also, analysis of phylogeny and taxonomic identification. And then finally, comparisons and statistics, the real ecological analyses at the end of the process. And so there are a number of considerations when you decide how you're going to analyze these data. And one of the first choices that you can make is you can use some available pipeline for analysis of these data, or you can develop your own pipeline. And I think anybody with the bioinformatic expertise available would probably prefer to have a customized pipeline for data analysis. Then, as I mentioned, a key point or purpose of the pipeline is to reduce the errors and biases, or at least to reduce their effects on the downstream analyses. And so there's some important things that can happen. And one is simply the choice of the binning threshold. And what I mean by this is deciding at what level you're going to discriminate between OTUs. So you could say that OTUs have to be 99% identical in order to be, considered to be part of one OTU. Or that threshold could be lowered. Commonly 97% identity is used. And this is really important because this will actually obviate the effects of many of the sequencing errors, since if there's only a single base change in a read, it's still going to get binned with the OTU in which it belongs. So that's a very important point here. Then once the binning is done, there are inevitably lots of singletons in these analyses. So you typically get very abundant OTUs, but inevitably you have this long tail of these very rare OTUs. And these may represent sequence errors or rare species, and there's really no way to know. So the singletons are a concern, and one of the things one can do is simply remove them from many of the analyses. So when we've done community comparisons, if we compare with and without the singletons, there's generally very little effect on the outcome of the analysis. And so getting rid of those singletons avoids having to worry about whether they may represent a lot of sequence errors. Also with 454 sequencing, homopolymers are very problematic. Again, this can be dealt with. What we commonly do is when we actually do the comparisons through alignments to decide to do the binning process, we just don't include any homopolymers. They're removed from those alignments, so they don't have any effect on the outcome. And then the last problem are chimeras occurring from the PCR, and that's something that I'm not aware of any good way to deal with. So it's very problematic with short reads to identify chimeras. Most of the chimera checking programs are actually made for full-length small subunit rRNA sequences. There are some algorithms now that are better for the short reads, but they generally, the ones I'm aware of at least, are not fast enough to really be practical for these large data sets that you get with thousands or routinely millions of reads to analyze. Okay, another important point is that the sequencing technology is constantly changing. And so these pipelines have to be revised to account for the various characteristics of the different kinds of sequencing. The occurrence of homopolymers or perhaps new problems that may occur with newer technologies. Finally, since there are so many options in how the data are analyzed, people are going to make different choices and the analyses will have different outcomes. And so this is something we have to keep in mind, and it makes it very difficult to compare between studies. So we have to be very cautious when we're trying to do this, because there could be big effects in what was found just based on how the data were analyzed. Okay, so now I want to turn to one aspect of community structure. And this is one that sort of came up in the previous talk, the idea of dispersion or variability of the community. And I'm going to use the same data set, the T-cell colitis induction experiment, to show the analysis of dispersion using this approach PermDisp. And the ordination really nicely demonstrates the principle. So each of these clouds of data for a treatment has a centroid. And the distance between the centroids is essentially the difference between the treatments. But another factor here is how tight the cluster is. So, for example, the green treatment, the RAG control mice, are a fairly tight cluster, whereas you can see the red are a much more diffuse cluster. And so these are more dispersed. And dispersion is a measure of this. And so when we analyze these data, what's plotted here is the dispersion, so that's the average dissimilarity for the entire group. And here we've actually plotted the time zero for the treated mice plus all of the control untreated mice. And so this is breaking it down for the rag mice versus the 45 rag mice. And what we see is a general trend of increasing dispersion, but it's not significant in the case of the rag mice. On the other hand, the 45 rag mice, it is significant. And so the latter two time points have greater dispersion, significantly greater, than where those communities started at the beginning of the experiment. So it's basically telling us that these communities are less stable over time and among mice. And so that's an important aspect of the community that I think it's really useful to be able to quantify in this way. And so to illustrate this in very basic terms, we can think of two very general types of change or difference between communities. And so you might have, in one case, this so-called positional effect, where basically the centroids move apart. So this could be over time or between treatments. Another possibility is dispersion alone could occur. And so you could see the centroid remaining in the same place, but the community could be more dispersed. And this would actually give you significant differences between these communities. But you'd need to do this analysis to see that it's actually due to this dispersion effect as opposed to positional effect. And then, of course, you can have a combination of the two. And so it's interesting, what we saw in the previous experiment is that in the case of the rag mice, we mainly had a change in the positional effect, whereas for the 45 rag mice, we also had a significant increase in dispersion that was affecting those communities. Okay, and what I want to do as I get towards the end, I want to talk about one additional approach for analyzing data. And first, I want to just show this tool, the Tree of Life. So this is a really useful online tool, and maybe some or many of you are familiar with this. It basically allows you to make a Tree of Life that can be customized in just about any way you want. So you can show whichever phyla you want, and you can show as much detail. You can collapse these down to phyla, as in most of these cases, or you can expand all the way down to the species level if you choose, so that you can focus on groups of particular interest. So here the proteobacteria have been expanded relative to the other phyla. Okay, so this is a nice tool, but what you can do with it then is you can plot data onto this tool and use this to visualize data. And so in this case, the data that we've plotted here is from indicator analysis. And this is another, I think, really useful approach from macroecology. And it's basically correlation of the OTUs with defined groups or with treatments. So it's looking for correlation. Which OTUs are really associated with particular treatments. The other thing about this Tree of Life is that what actually happens is your tree that you construct resides online. So you can actually have a link to this in a publication, and you can have a massive amount of data available to anybody who wants to inspect it. But then you can, of course, also produce figures that show specific points that you want to make. And so I just want to show this indicator analysis in a little more detail. And so here, I'm not going to talk about all of the data, but I just want to focus on the second, third, fourth, and fifth rows of data. And I should say that this is not actually animal microbiome data. I really wanted to get this Tree of Life figure with indicator analysis for the colitis induction experiment, but the postdoc in my lab who's doing that work was really sick last week. So what I've used here is a figure from some of our analyses of forest soil microbial communities. But it makes the point that I want to make just as well. So these four concentric rings then are showing data from 4 different treatments. within an experiment. And the size of these bubbles is proportional to the relative abundance of each taxon that's shown. And then the color, where indicated, so where the bubbles are colored, shows that these taxa are significant indicators. Okay, so for example, here just kind of focusing on the proteobacteria, if we look here at the gamma proteobacteria, they are not only very abundant, but a significant indicator for this particular treatment, whereas they're much less abundant in the other treatments, although still indicators. On the other hand, some of the dots are black, so you can have a relatively abundant taxon, which is not an indicator. And you can have taxa that are not very abundant be indicators as well. But anyway, this is a way to inspect the data and to put it into a phylogenetic context, which I think is really useful for when you want to analyze these data. Okay, so what I lastly want to do is to sort of summarize many of the considerations that I've discussed by showing you sort of a quality ranking of what can be inferred from the amplicon sequencing approach to community analysis. And so this takes into account the impacts of the various biases that I mentioned. And so there are a number of things that this analysis can do very well. It's very good at showing the presence of abundant populations, and it's very good for determining the similarities of communities within a study, as well comparing relative abundances within a study, and measuring evenness, which is an important component of diversity. So evenness is basically the extent to which the individual OTUs are distributed. So if you have a dominant OTU, you have an uneven community. And of course, that can be a very important aspect of a community. And so the amplicon sequencing approaches are good at telling us about evenness. On the other hand, these approaches are not so good for comparisons of communities among studies or meta-analyses. Okay, so for the reasons I said, it's hard to compare between studies given different methodologies. Also, the methods are not very good for determining the presence of rare populations, because this gets into this issue of experimental artifacts such as sequence errors, which can't really be well distinguished from true rare populations. And these approaches are very bad for making any conclusions about the absence of particular populations in a community, or for looking at the richness component of diversity. And so I would argue that today we still don't have a good way to determine richness. So when we talk about 1,000 species in the human gut, we know that there are that many, but we certainly don't know how many others might be there. And the problem again is that when we start to look at the rare species or the rare OTUs, we simply can't draw the line between what are true populations versus what are artifacts of our analyses. Okay, so I'll stop here. I want to give thanks to a number of people who contributed to this work, especially two postdocs in the lab, Pedro Dimitriou and Martin Hartmann. And merci.